Hello, I'm John Coffin. I'm on the faculty at Tufts Medical School in Boston, but I'm very closely associated with the HIV Dynamics and Replication program here in Frederick, Maryland. And I and my colleague Stephen Hughes are here to discuss a recent paper that we published in JCI Insights on the origin of clonal populations of HIV-infected cells in patients soon after they are infected with HIV. These clonal populations of cells, as we have shown earlier and others have shown as well, are what prevent an individual infected with HIV from being cured because no matter how effectively the current drugs, and are very effective, no matter how effectively they block infection of new cells, they cannot deal with the cells that are already infected, live for a long time, and indeed even keep on growing in individual cells, leading to large populations of cells that carry viruses that can then rebound very quickly once the virus therapy has been stopped. At this point, I'd like to introduce you to my colleague and collaborator and friend, Stephen Hughes, who will introduce himself and present the results of our study. Steve.
Thank you, John. I'm Steve Hughes from the HIV Dynamics and Replication program, and together with my colleague John Coffin, we represent a much larger collaborative effort. And in just a moment, I'll tell you about what we found in our study. As my colleague John Coffin just told you, effective antiviral therapy blocks HIV replication, but does not cure HIV infections. If therapy is stopped, viral replication resumes. CD4 plus helper cells are the principal targets for HIV infection in patients. In uninfected people, CD4 plus T cells can clonally expand in response to specific antigens or cytokines. As you can see on the first slide, in HIV-infected people, a small fraction of the HIV-infected CD4 plus T cells clonally expands and persists even in the face of effective antiretroviral therapy that blocks HIV replication. Infected clones are able to survive because only a small fraction of the cells in the clones are making viral RNA and proteins at any one time. Although in patients on therapy, most of the HIV-infected CD4 plus T cells carry DNA copies of the HIV genome, which are called proviruses. Most of these proviruses are defective. However, there are large clones of infected cells that carry fully infectious proviruses and release infectious virus into the blood. These clones are an important part of the reservoir of infected cells that has made it impossible to cure HIV infections with the available antiviral drug therapies. On the next slide, we consider the question of whether there is ongoing replication in HIV-infected individuals on effective antiviral therapy. Despite claims to the contrary, there is no evidence that there is ongoing replication in patients whose therapy is effective. HIV replication is error-prone, and viral replication leads to the accumulation of mutations in the HIV genome. In people, most HIV infections are initiated by a single provirus. That means that shortly after a person is infected, all of the viral genomes are virtually identical. And you can see that in this slide by looking at the open triangles for the 5 patients. These are the sequences from the viruses that are present before antiretroviral therapy is initiated. This makes it easy, if people are treated within the first few weeks after they are infected, to see if viral mutations arise while these patients are on therapy. Slides compares the viral sequences that were present initially, as I already mentioned, to those that are found after 2 to 3 years of successful therapy. Those are the filled-in triangles. There is no evidence of any new viral mutations that arose while these patients were on therapy, which means that there's no evidence that there is ongoing viral replication when these people were on therapy. We go to the next slide. We know that the reservoir of HIV-infected cells is established shortly after someone is infected with HIV. If, as we suggested earlier, clones of HIV-infected T cells make up an important part of the reservoir. Clones of infected cells should arise soon after a person is infected with HIV. It is usually impossible to know exactly when someone was infected with HIV. However, as was first pointed out by Fiebig and his colleagues, the progressive appearance of viral RNA, viral proteins, and the immune response of the host can be used to estimate the time since the initial infection. As we will show you in the next few slides, the period we are most interested in is Fiebig 4-5, which is approximately a month after the initial infection. In the next slide, this slide shows that the viral DNA load, the number of viral DNA copies per million cells, in the patients both before therapy was initiated and after 2 to 3 years of successful therapy. As you can see, and as we expected, blocking new viral infections reduced the viral load in all the patients. Many of the newly infected cells die shortly after they are infected, and the therapy prevented additional cells from being infected. So you see that the viral load is higher in the samples taken pre-therapy on blue than in the on-therapy samples in orange. In the next slide, we consider the problem of integration. HIV DNA integration is not random, but most proviruses can be integrated into millions of different sites in the host genome. If an HIV-infected cell clonally expands, all of the daughter cells will have the provirus integrated in exactly the same place in the host genome. If we can show that in a sample taken from a patient, there are multiple cells that have exactly the same integration site, we know that these cells are part of a clone of infected cells. Here we show that we can determine the integration sites of HIV proviruses in cells taken from recently infected individuals, both before the patients went on therapy, that's shown in blue, and after the patients have been on therapy for 2 to 3 years, which is shown in orange. In the next slide, we looked for identical integration sites, that is to say clones of infected cells, in samples taken both before and after the patients went on therapy. Because there is a large background of integration sites in newly infected cells in the pre-therapy samples, it was much easier for us to detect clones of infected cells in the on-therapy samples. We did not detect any clones of infected cells in the pre-therapy samples from those patients who were analyzed at Fiebig 3-4. And those are the open purple circles at the left side of the slide. However, we did detect clones of infected cells in the on-therapy samples from these same patients, and that's shown in orange again at the left. Because there was no ongoing viral replication on therapy, it is likely that these clones derived from cells that were infected before the patients went on therapy. Presumably, clones of infected cells were present, but the clones were still too small for us to detect in the Fiebig 3-4 pre-therapy samples. By Fiebig 4-5, about a month after the initial infection, there were clones of infected cells in the pre-therapy samples that had grown large enough for us to detect approximately 100,000 infected cells in the clone. Some of the clones we detected in the pre-therapy samples persisted, and the same clones could be detected in samples taken after 2 or 3 years on therapy. So, in conclusion, we think we've shown that HIV-infected cells clonally expand early, which is what we would expect if they are an important part of the reservoir that makes it impossible to cure HIV with current antiretroviral therapy. Thank you very much for your interest in our study.
And thank you for your attention.